we saw an interesting video of some peristalsis. Today we're going to see a couple more videos about the, we'll say, the other end of the digestive tract. Once we get into the large intestine, into the colon, we'll see some videos from inside there, and then we'll talk about that last little bit of control that you're going to regain over the digestive system at the very end. And then after our break today, we are going to get a bit of a start on the urinary system, so we're in good shape for next week to really have a good handle on how the kidneys are going to work, one of the most important systems that we have in the body. I know I've said that about all of them, but no, kidneys are very, very important too. But before we get to that, we do want to talk a little bit about the absorption that's going to be happening from the small intestine. Yesterday we looked at the structure of the small intestine, and what did we say about that structure of the small intestine? What was it all about? Surface area. Hey, we can remember that. Surface area. Surface area, surface area, surface area. Surface area for not only the chemical digestion, so that we're able to have all of those macromolecules come into contact with those enzymes, but then of course the surface area for absorption. So if you remember back to yesterday, we talked about this structure of the villus, and at the center of this finger-like projection, we've got our capillary bed that is going to be at the center, as well as this specialized lymphatic capillary that we call a lacteal. We call it a lacteal because the lymph that is going to be found in that specialized lymphatic capillary takes on a bit of a kind of a milky color, and that's because of the type of nutrient that is going to most often be absorbed into this lacteal, which, as we're going to see, is one of our breakdown products of lipid digestion, so fat digestion. And so it gives it kind of this milkish color, and so that's where that name lacteal comes from. All right, so this is the structure that we're talking about here and where this absorption is going to be happening. And so what we find then is that in order to have absorption, we have to talk about the different types of breakdown products. And so these different types of end products of digestion are going to be absorbed through different mechanisms, but all of these, the ones that you see here, are going to end up directly moving into the blood capillary. Okay, so things like our glucose and our galactose, these monosaccharides, where did they come from? What did they used to be? Carbohydrates, starches, right? Big carbohydrate molecules. Then we use those amylases to be able to break them down into their building blocks. And so they're then going to be able to be absorbed through those absorptive cells. What are these structures here along this apical surface of these absorptive cells? Those are the microvilli again, and this is a nice way of drawing it, so you can see it's just kind of these ripples or these undulations in the cell membrane. They're not cilia, they're not those hair-like processes that we talked about for other types of tissue. And so we're able to move these relatively small subunits now right through the absorptive cells and then into the blood capillary, where then it can be sent over to the liver and then further processed, or then sent further back to the heart and then all around the body. Similar for amino acids. What are amino acids the building blocks of? Proteins. Again, we were talking about very small building blocks when it comes to fructose or glucose. Amino acids. Dipeptides just means two amino acids still linked together. Tripeptides, three amino acids linked together. But those are all small enough to be absorbed pretty easily, again, into our absorptive cells, and then right out the other side into our blood capillaries. And then finally, of course, our fatty acids, where are they coming from? Lipids is the other major macromolecule that we have left, so our fats. And so we have our short chain fatty acids that are going to be absorbed, again, directly through the cell and then into our blood capillary. Now, short chain fatty acids are one component, one end product of chemical digestion of lipids. But there's another kind of major category, or a couple of categories of components that we don't see on this image here, and that's because they're much too large. In order for substances to be absorbed into the blood capillary across that capillary wall, what do we know about that capillary wall? Is what? Size? Is that what you said? I can't hear you. I think getting closer, you're getting quiet. Okay, sinusoid. Okay, so So sinusoids are going to be in the liver. So we find those sinusoids in the liver. These are going to be continuous capillaries that we find here. And so these continuous capillaries are going to be how permeable? Not as permeable as things like sinusoids or as fenestrated capillaries. Still reasonably permeable, but not as permeable as some of these other types of capillaries. And so the capillary wall is semi-permeable, and it's really all about size. So if substances are too large, then not only are they not going to be able to get out of the capillary, as we talked about before, they're not going to be able to get into the capillary either. Now things like glucose and fructose, amino acids, these short-chain fatty acids, they are small enough to be able to get across that semi-permeable membrane of the capillary and move into the bloodstream. But two of the breakdown products of lipids are much, much too large, and they're also going to be hydrophobic. And these are going to have a much, much harder time moving directly into the bloodstream from the lumen of our small intestine. And so what we're talking about here are long-chain fatty acids. If you think back to your biochem class from high school, chemistry class from high school, think about these long-chain fatty acids. What are they? Well, they're just lots of carbons. Carbon, 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 carbon. Sometimes dozens and dozens and dozens of carbons all in a big long line. Those are long-chain fatty acids. Well, it turns out that they're just very big. And so if you're getting too big, then that's going to be too hard to get that molecule into or across that capillary wall and into the bloodstream. And a lot of times these long-chain fatty acids and monoglycerides, which are another breakdown product of lipids, are going to be hydrophobic. So if they're hydrophobic, what do they not want to dissolve in? Water. Well, what is blood plasma mostly made up of? Water. Okay, so they're not going to easily or really want to be able to dissolve in that water to be absorbed very well. And so we have to come up with some other mechanism. And so what the body has come up with is that we're going to take these large hydrophobic molecules and we're going to package them together inside the absorptive cell, inside this phospholipid and cholesterol package. It's almost like a mini cell membrane. It doesn't have all the proteins, it doesn't have all the components that a normal cell membrane does, but it's got a bunch of phospholipids, it's got some cholesterol in there, and it's going to form this nice little package around these building blocks of those fats. And so this is then how we're going to get around the hydrophobicity of these components. If we can package them inside this little, almost like a cell membrane package, well then the outside of what we call this chylomicron can interact with water, and then we're going to be able to transport it much more easily. The problem with this, of course, is that if you're packaging all of these relatively large breakdown products inside an even bigger, almost cell membrane-like package, your whole chylomicron, your whole big package here is now way too big to be able to get across that capillary wall. So where else could we send it? Well, we can send it to the other type of capillary that is found at the center of our villus, which of course is a lacteal. And if you think back to your studies looking at the lymphatic system, we talked about how the cells of those lymphatic capillaries, they overlap each other, and there's a relatively large space in that kind of one-way valve or one-way overlap of those cells. And it will allow drainage not only of proteins from the interstitial space, which are large and we have to get rid of them, so that's why that kind of more permeability of those lymphatic capillaries is helpful. But now also here, these specialized lymphatic capillaries will allow for these chylomicrons, which contain these breakdown products of fat metabolism, into the lymphatic system. And then from the lymphatic system, where does that lymph eventually end up? Back into the bloodstream, right? So we dump it back into the bloodstream, and that's then going to be able to get us these long-chain fatty acids and those monoglycerides into the bloodstream so that we can have them processed further by the liver eventually, and then used by our normal body cells. So other than these large breakdown products of lipid metabolism, we're going to be able to get all of our nutrients absorbed right through the absorptive cells and into the bloodstream. It's just that for these ones, we need a bit of a different pathway through that, using that chylomicron and then into the lacteal. having absorption of nutrients. Fantastic. What about water absorption? This is a big one because as we've seen, and we talked about all the way along, and we've held up liter water bottles and said, look at this, you make a liter of spit every day, you make a liter of bile every day, you make a liter and a half of gastric juice. So you can go through and start adding up all of the volumes of fluid that you are secreting into your digestive system. And it turns out that over the course of a day, you're secreting about 9 liters of water. Right? Well, initially, where is all of that fluid originating from? Right? The saliva that you're creating, where did the fluid from the saliva come from? Yeah, your salivary glands. Where did the salivary glands get the fluid, get the water that it needs to make saliva? From the blood. Where did your cells in the line that line your stomach get the water that it needs in order to be able to produce our gastric juice? From the blood. Where did the intestines get all of it? Where did the pancreas get all the water? Where did everything get all of the water that it needs to make the secretions that it makes? Well, from the blood. How much blood do you have? 5 liters of blood. You're secreting 9 liters of water every day over the course of the day into your digestive system. So obviously you're not leaving those 9 liters of water in your bloodstream. Now I should say, okay, you're taking in some of this 9 liters as a couple of liters that you're drinking for your coffee or water or whatever. But there's 9 liters that's going to end up in your digestive system every day. You're obviously not leaving 9 liters in your digestive system to travel all the way through. Otherwise, what would you be experiencing every day? What's that? Yeah, diarrhea, right? Then you'd be losing 9 liters of water, right, out the other end. Well, we're obviously not doing that unless that's kind of gastrointestinal issue. We're going to have to get this back, right? And so we're going to have to reabsorb the vast majority of all of this fluid that we've either taken in or secreted as part of our saliva and our gastric juice and our intestinal juice and our pancreatic juice and our bile and all these fluids that we're secreting, we're then going to have to reabsorb them back. And so most of that does get reabsorbed simply by osmosis. So again, as we're moving those nutrients and as we're moving those ions, absorbing those ions into the blood, well then water is going to be able to follow. And again, most of this reabsorption happens in the small intestine. By the time we then get to the large intestine, we've reabsorbed already about 8 of those 9 liters. Okay, so we've got like 8 out of those 9 have come back into the bloodstream already. That leaves us about a liter of water entering into the large intestine. Well, again, a liter of water, you're not losing a liter of water in your feces every day. And so one of the jobs, as we will see, of the large intestine is then going to be to reabsorb most of that last liter or so of fluid, so that by the end of the whole pathway, you're left with about 100 milliliters. And so you start with 9 liters, 9,000 milliliters being moved into the lumen of your digestive tract at some point along the way. And by the very end, when you are going to become responsible once again for controlling what goes on and the timing of what goes on, you're left with about 100 milliliters. So we do a pretty good job of getting the vast majority of that water back. Now, we don't want to get all of the water back. We don't want the feces to be completely dry because that's going to be problematic for our intestines, for our large intestine as well. So we need it to be reasonably soft to allow it to continue to move through the distal portion of the large intestine to the outside of the body. But we also don't want it to be too wet, otherwise you're just going to be losing electrolytes and sugar. All right, so keep this in mind that we are going to have to use or rely on this large intestine to reabsorb some of our water, but the vast majority of the reabsorption is happening in the small intestine. Great, all right, so let's move on then to talk about the large intestine or the colon. So you can use these two terms interchangeably. Sometimes people will talk about this as being the bowel. So you can have the bowel, the colon, the large intestine. They all mean the same thing. When we look at the large intestine, there's a couple of interesting features that we want to keep in mind before we start labeling these different parts. It's about 1.5 meters long. Okay, so again, not nearly as long as the small intestine. So we said yesterday, it's the diameter of the tube that is determining whether it's the small intestine or the large intestine. So this is much larger in diameter, about 3 times-ish more in diameter than what we see for the small intestine. And it's going to extend from that ileocecal valve, or that ileocecal sphincter, all the way down to the anal opening. Again, you still don't have any voluntary control over what's going on in the large intestine until that very, very last couple of centimeters, where again we'll talk about that voluntary control being regained once again. We talked earlier on about the mesocolon being responsible for holding it in place, so that again you can have this kind of 3-dimensional structure here and organization where there's going to be some pretty hard turns, if you want, some 90-degree turns in this pathway, and that's all going to be held in place with that mesocolon. So when we look then at the proximal portion of the large intestine, we see that this is the cecum. And so the cecum, again, when you talk about the connection point between the small intestine and the large intestine, what was that valve called? The ileocecal valve, or the ileocecal sphincter. Again, as we said yesterday, ileocecal, that's telling you that it's the connection point between the ileum of the small intestine and here the cecum of the large intestine. Okay, so again, that helps you remember, hopefully, where that connection point happens. So here you can see a section where they've opened up in the drawing this area of the cecum, and hanging off of the cecum is where we find the appendix. Okay, so the appendix is kind of an interesting component of the digestive system. Here you can see where the cecum has been cut open, so very similar to what they've done here in the drawing, is what they've done here in the cadaver. So this is kind of our little window that they've cut so you can see inside this cecum. There's a nice little sphincter right here. What is that sphincter? Yeah, that's our ileocecal valve, or our ileocecal sphincter. So if you were to send the camera back through that opening, you would end up back into the ileum of the small intestine. So you kind of see that pathway then as that chyme is going to be moving, or what's left of the chyme is going to be moving towards us into the cecum, and then as we'll see, it's going to be working its way in a superior direction as it works its way up and then across the abdominal cavity. You can see right here, kind of hanging off the side there, is the appendix itself. So it extends out from the cecum. And for a long time, most people thought that the appendix was really kind of not useful for very much. You can pop it out, you can leave it in, sometimes it gets inflamed. There's oftentimes going to be associated with gut infections, but they didn't really understand kind of like what was it for. Maybe it's just a leftover from earlier in evolution. Who knows why it's there, but it doesn't seem to have very much value. More and more, though, what they're finding is that because of the large amount of lymphatic tissue that we find in the appendix, and the amount of bacteria, healthy, good bacteria, that live in the appendix, that the appendix is actually a pretty important kind of training site, if you want, for your immune system. And so what they think is happening is, again, a couple of theories, and there's kind of more or less evidence for some of these ideas, but it's thought that the appendix, one, can act as a reservoir for healthy bacteria. So that we want to be able to make sure that if there's some kind of massive die-off event in the large intestine that's wiping out large populations of our normal healthy bacteria, what we call the normal flora, right? We talked about this in Dr. Stewart's class. Yes, fantastic. So you know then about this normal flora that lives on and in us, and a large proportion of that lives in the large intestine. So it's thought then that by, if you have a massive die-off, if you eat some kind of gastrointestinal toxin or whatever, and so your body tries to wash that out, and along the way it washes out a whole bunch of bacteria, that the appendix can act as a reservoir for those healthy bacteria. It's also thought that because, again, of all this lymphatic cells that are going to be found in the appendix also, that it is helpful for training your immune system to not attack the healthy bacteria that are living in your intestine. Right? Because if you think, wait a minute, how are those bacteria able to live there? Shouldn't there be a massive immune response that's happening all the time to try to rid your body of all of these thousands and thousands and thousands of bacteria that live there? And we think, well, yeah, that would, why isn't the immune system fighting it off? And it's thought that maybe it has something to do with the lymphatic tissue that's in the appendix, and the bacteria kind of coming to an understanding where, listen, we're going to live here, you're going to live over there, we're going to make sure that we stay living in the large intestine and are not migrating into the small intestine. Okay? Because if we get bacterial growth in the small intestine, now you're starting to have kind of more major infections. There should not be very many bacteria living in that small intestine. And so it's helpful to kind of train the lymphatic system to know what to look for if those bacteria end up in a place that they're not, but then to kind of keep those bacteria under control so they're not spreading too much from that large intestine. So more and more is those active areas of research on the appendix and how it might not be as totally useless as people once thought that it was. All right, so lots of bacteria then are going to be living in this large intestine, and there's more and more research all the time on the link between the health of that populations, those populations of bacteria, and overall health in the body as well. There's some evidence to suggest that there's somehow kind of communication that's happening between those bacteria and even nervous signals that are being sent to the brain. So lots of new interesting research that's happening about the health of your gut flora. But working our way superiorly from the cecum, we move into the ascending colon. Why do you think this is called the ascending colon? It's moving, right? It's moving in this superior direction. And so again, this is a little bit counterintuitive when we think about the movement of really what's kind of essentially fluid, but it is going to be moving in this ascending direction until it reaches this kind of 90-degree turn. And then we have the transverse colon. Why is it called the transverse colon? It's going across, it's traversing your abdominal cavity, exactly right. And so it's going to be traversing from one side to the other, and then we're going to have the, oh, surprise, surprise, the descending colon as we're working our way down then to the sigmoid colon. So the sigmoid colon is named because it's sort of a sideways S shape, and so this kind of sideways S-shaped area is the sigmoid colon, which takes us to the rectum, which is going to have a lot of smooth muscle in the wall. This is going to be the major muscle that is going to help move the feces towards the outside of the body. And then we have the anal canal. And so it's in the anal canal where we're going to have our final two sphincters to be able to close off this portion of this pathway, and then brings to an end this idea of all of the sections of the digestive tract being organized into discrete areas. Right? So we started with our esophagus. We said the esophagus has a sphincter at either end. Then we talked about the stomach, and the stomach has a sphincter at either end. Then the small intestine has a sphincter at either end, and now the large intestine has a sphincter at either end. So we can essentially isolate each of the different sections of the GI tract with these sphincters, including the very end at the anal canal here. A couple of other features to keep in mind are these 90-degree turns that we find. So this is called the right colic flexure. Okay, so right colic flexure because why? It's on the right side, and it's the colon. Exactly right. So this used to be called the hepatic flexure, and that's of course because it's close by to the liver, so the hepatic flexure. But it has nothing to do with the liver, and so a lot of times people were getting confused. Well, the hepatic flexure, that's something to do with the liver itself, but it's not. It has everything to do with the colon. It's just that it was close by to the liver, just like this was called the left colic flexure. It used to be called the splenic flexure because it was close by to the spleen, but it has nothing to do with the spleen. So they've changed the name now to simply say the right colic flexure and the left colic flexure, so that we know that we're making these kind of 90-degree turns at those areas. Right. This then brings us to talking about the distal end of the colon itself, this large intestine. So this distal end is going to essentially begin with the rectum, and this structure, this organization is going to become important in a few minutes when we talk about defecation and how are we actually able to, first of all, sense and then be able to control when defecation is going to be happening. So again, the rectum, the distal end of the colon here, about 20 centimeters or so in length, anterior to the sacrum and to the coccyx. So we know that again, when you're talking, try to figure out where it's located. It is found anterior to that very distal end or inferior end of the spinal column. And the rectum then is going to have a lot of muscle in the wall, a lot of smooth muscle in this wall, much of it organized in a lengthwise fashion, so a longitudinal fashion. So as it contracts, it's going to be able to squeeze the feces down towards this anal canal. The anal canal itself is the last kind of 2 to 3 centimeters or so of the GI tract. And the big idea about the anal canal are the 2 sphincters that are going to be found there. One of them, and again, it's important that we kind of recognize this organization of internal versus external sphincters, because we're going to see exactly the same pattern of organization of an internal and an external sphincter when we talk about the urinary system coming up. And there's an internal and an external urethral sphincter as well. And the process of being able to control urination is exactly the same as what we're going to talk about here. So again, it'll be a bit of review once we get to that urinary system. So we start with the internal anal sphincter, and the internal anal sphincter is a ring of smooth muscle. So again, it's smooth muscle, it's involuntarily controlled. You don't decide when this contracts, when this relaxes. What you do control, though, as I said, is the very, very last moment of this whole process. You lost control when the bolus was in your esophagus, and now you're going to gain the very last moment of control right here at the very end with our external anal sphincter, which is now a ring of skeletal muscle and is under voluntary control. So hopefully, as I said, all of you right now should be keeping that external anal sphincter nice and tightly closed. Regardless of what the internal one is doing, you can't control that, so that's fine. But that external one, hopefully you've got some pretty good voluntary contraction that's happening on that sphincter. Okay, so this is going to be the organization of the very distal end of the large intestine, and we're going to come back and talk especially about these 2 sphincters when we get to looking at defecation in a few minutes. Now what we want to look at is the histology and what's happening in terms of the mechanical digestion and the absorption, so that we can then get to, well, what is feces and then how does it get out of your body. So when we look at the inner lining of the large intestine, we find that the mucosa looks significantly different than what we saw for our small intestine. For our small intestine, what was the whole point? Surface area. It was all about surface area. So you had those circular folds, you had those villi, you had those microvilli. It was all about increasing the surface area for digestion and absorption. But now, by the time we've gotten to the large intestine, what have we already mostly done? Absorb everything. We've absorbed the vast majority of the things that we needed to, and so there's not really a need to maximize surface area anymore. What we're doing in the large intestine is essentially going to be fine-tuning the last little bit of absorption that needs to happen. The last little bit of water, the last little bit of electrolytes that need to be pulled out, and giving a nice space for the bacteria to be able to grow. So it turns out that we don't have any villi, no circular folds, so the mucosal layer is going to be relatively smooth. We have these little indentations here and there. That gives a nice little pocket for bacteria to be able to grow, so they're not going to be totally washed away all the time. Okay? But it's relatively smooth along the inner surface, and we'll see a couple of images and videos of that, of an actual inside of a colon in a couple of minutes, and show you then what that difference looks like. Mostly, you're going to have absorptive cells, and we're going to be absorbing most of that last little bit of water that we need to, as well as some goblet cells that's going to, of course, release some mucus, make sure that the feces, the leftover undigested food, is able to kind of continue to work its way along. It isn't going to get caught along the way. The rest of our components, the rest of the 3 layers of our digestive tract here, are going to be the same, and so there's nothing really more to say about that, other than this muscle layer, which is organized in a bit of a different fashion. So still 2 layers, we still have 2 layers, a circular organization and a longitudinal organization, like we saw in the small intestine, like we saw in the esophagus. But now what we find is that the muscularis layer, especially this longitudinal layer, is going to have these kind of thick bands of this smooth muscle, these longitudinal fibers. And that's called the tenia coli. So you can see this kind of bands that they're drawing here. That's the tenia coli, these outer thickened layers that essentially kind of scrunch up the large intestine into these little pockets. So it's almost like you take a like a long, I don't know, hose or a long sock or something, and then you just kind of like scrunch it all in, and then you've got all these kind of little pockets, all these little kind of bulges all the way along of this large intestine. And so each one then of these little pockets, of these little pouches, if you want, is called a hostrum. So multiple hostrum are called hostra, so that's just the plural version of one individual hostrum. The yellow here in this drawing is supposed to represent to you that there's some adipose tissue associated with the outside of the colon, but the important idea is that these bands of tissue, or these thickened areas of that longitudinal smooth muscle that helps to kind of gather together or scrunch up that colon into those hostra. Of course, there's that muscularis layer then, that I should say serosa, not muscularis, sorry. What does it say in your notes? It says muscularis, you can delete that and put serosa, because that's of course the outermost layer of those four. Sorry about that, that's a type. Same 4 layers as always. Muscularis is the interesting one, and then serosa, or just connective tissue on the outside. All right, so let's take a look then at the inside of the colon to see the difference between the colon and the small intestine. Remember yesterday we saw some of those images that were showing you those circular folds, that the circular fold itself looked a little bit kind of fuzzy or furry because it had those villi on them. But now, as we send a camera up into the colon, we see a couple of these different features now that we're talking about. So this first video is going to show us a view from the cecum. So the camera has worked its way from the rectum all the way up around the entire length and is now in the cecum itself. And so we're able to see, as again, a little bit of the chyme that's in here. You can see the smoothness of the walls, so we're not seeing all of those villi. This is the opening into the appendix. So you can see the opening to the appendix there, but the smoothness of the wall is kind of one of those key features. Then if we look down here, in this view, let me just pause there for a second. In this view, the camera again is going to be beginning in the cecum, but actually has pushed backwards through that ileocecal valve. So what we're able to see here is actually the very distal end. This is the very distal end of the ileum of the small intestine, and then we'll see that the camera pulls itself back out of that valve and then ends up into the cecum and then into the ascending colon in just a second. Okay, so that's what we're looking at. We're looking backwards now. As the camera pulls back, we'll be able to see the ileocecal valve. There's some of that. Okay, so this is the ileum all the way back into the small intestine. And then as the camera pulls back, there's the ileocecal valve that you can see right there. This is the cecum down in here. And then the camera is working its way kind of in an ascending direction, coming backwards. So you can see all of these little ridges. What are all of these little pouches called here? Starts with an H. The hostra, exactly right. So each of these are the individual little hostra, and once again, you can see that smooth surface of the inner lining of the large intestine. I know you're all wanting to watch that again, so we'll watch that again. So you can see again backwards into the small intestine, and all this movement that's happening. Right, we're talking about all this contraction that's taking place. That's our peristalsis that we were looking at the other day, yesterday. And so as we pull backwards. Again, that's the cecum, the ileocecal valve, and then you can see this is our ascending colon and all of those individual hostra. And you can see all those pouches then that are going to be containing all of that chyme. So you wonder, how is it then that we're able to move the contents, this chyme, and what's eventually going to become feces, in an ascending direction, and then once it gets into the descending, why is it all just not pouring down to the bottom? Well, it's because it's contained in these little pouches. And so in order to move the contents from one hostrum to the next, we are going to have some contraction of each hostrum. And we're going to have to kind of churn it around a little bit to be able to get the movement of the chyme happening kind of along in sort of segmental fashion from one end to the other. And so that then brings us to our mechanical activity. Again, as always, there's going to be mechanical digestion and chemical digestion that's happening in varying degrees. Okay, so when we talk about this mechanical digestion in the large intestine, we see that it's actually not all that active all the time. The stomach is very active, the small intestine is very active, but the large intestine is not nearly as active. The contractions are much more sluggish, so we don't see kind of the nice, smooth contractions that we were talking about yesterday. We saw that video of the waves of peristalsis that were nice and regular and nice and smooth. Here it's a little bit more kind of short-lived. It's a little bit more kind of haphazard in terms of when these contractions are going to take place. But there are generally three kinds of contractions that happen in the large intestine. Yes, there's the peristalsis that takes place, not nearly as often in the large intestine as what we saw in the small intestine. But then we have haustral churning and then mass peristalsis. So haustral churning, we'll start with haustral churning. What do you think is going on here? Churning of the haustrum. Yeah, exactly right. So there's going to be some kind of local movement of the haustrum that will, again, doesn't occur very often, every 30 minutes or so. We're going to have these slow segmenting movements. That's what we mean by sluggish, so it takes longer for these contractions to happen. But that's what's going to move, and we should say the chyme, not the food there, move that chyme from one haustrum to the next. So you're kind of moving it slowly along. We're going to do a little bit of absorption in this little pouch, and then we're going to like into the next little pouch, and then we kind of move around and have some absorption, and then we're into the next one. So we're kind of moving from one to the next to the next, but again, relatively slowly, only every 30 minutes or so are you going to have kind of this movement from one haustrum to the next. Mass peristalsis is a little bit different, because mass peristalsis is going to occur when you really are ready to make some room for new chyme that's coming in. When you're not eating anything and there's no new chyme coming from the stomach into the small intestine or from the small intestine into the large intestine, then that's fine. Haustral churning can kind of do its thing, and we just slowly kind of move the contents from one haustrum to the next along the pathway. But at some points throughout the day, there's going to be a big movement of chyme. You want to kind of clear out the earlier parts of the pathway to make room for new chyme that's coming in. Right? When does that normally happen? A couple of times a day, making room for new chyme. When you're eating something, yeah, when you have something to eat, right? When you eat something and you fill up your stomach, well, now you want to make sure that whatever chyme is left in the small intestine, it's had time to kind of do its thing. We need to move that along, right, and make some space for this new chyme that's about to enter in. And then, if you're making space in the small intestine, well, you better be making space in the large intestine. That you kind of have to move everything down the line further, right? And so that's where mass peristalsis comes into play. Mass peristalsis happens on average 3 to 4 times per day, very often during or just after eating a meal. And this is going to be activated by a specific reflex that we'll talk about in just a second, this gastrocolic reflex. Gastrocolic. Gastro means stomach, colic, colon. So there's going to be some link between what's going on in the stomach and what's going on in the colon. So keep that in mind as we go along. And so there's going to be these kind of large sections of the wall of the colon that are all going to be kind of contracting together and kind of pushing the contents of the colon down towards the rectum. Because as I said, if you're going to be eating a meal at that end and adding more chyme in at this end of the pathway, well, then you got to make space along the way. And so we're going to make space along the way by this mass peristalsis. So we begin kind of in the middle of the transverse colon, and then all of the contents of the colon are going to be then pushed down towards the rectum to work its way out so that we have space for the new chyme that's coming in. So if you've ever been to your family dinners or whatever, and Grandpa goes to the bathroom with a big magazine, takes his National Geographic to the bathroom right after the big dinner. Like, why do you always go to the bathroom after dinner? Or like my dad, why do you always go to the bathroom right when dinner is about to happen? Say, okay, dinner time, you're right there. And off he goes. What are you doing? Okay, well, it's not their fault, right? This is just mass peristalsis. We're just making space for the new chyme that's going to be coming in. So don't be too hard on Grandpa. Don't be too hard on my dad. Like, you can't help it. It's a reflex, and so kind of have to just give them the time, it's fine, potatoes will still be warm when you get back, and then there'll be lots of space to have that new kind coming in. So let's talk about these two reflexes then that are going to control this movement. So as we said, we're trying to make space. You got to make space at the very end, but you also have to make space in the middle as well. And so there's two important reflexes that are going to help to control this contraction. And so the first is the gastroileal reflex. Gastroileal. Stomach and ileum. Look at that. It's telling you exactly the two components of this reflex. So gastrin, oh my gosh, not gastrin again. Yes, gastrin again. Gastrin that's released from the stomach. What did we say gastrin was going to do for us? It increases our gastric motility and secretion. That was our hormone control of gastric functions. We said that when there's chyme in the stomach, then you're going to have, or bolus that has arrived in the stomach, you're then going to have the release of gastrin. Gastrin increases the motility and the secretion of the stomach. That's just going to help to kind of get things going in that gastric phase of gastric function. And not only is the gastrin then going to act on the stomach to kind of really ramp up motility and secretion in the stomach, because it's a hormone, it's going to then get released into the bloodstream. And so, yes, it'll work on the stomach. It will also relax the ileocecal sphincter. So why would we want to have the ileocecal sphincter relaxed? What is that going to do? Got a whole bunch of new chyme in the stomach, and now that same stimulus is going to cause a relaxation of the ileocecal sphincter. Where is the ileocecal sphincter? Between the small and the large intestine. So why would we want to have that relax? Yeah. Because we want to move whatever chyme is left in the small intestine. We want to get it more easily moving into the large intestine. Make space for that new chyme that's coming in from the stomach. Great. Then we're going to have our gastrocolic reflex, as we said a few minutes ago, stomach and colon. That is now what's going to cause our mass peristalsis to happen. If you've got more chyme coming in from the small intestine, you better make some space in the large intestine, and so we have our mass peristalsis then that's going to move the contents of the large intestine down towards that rectum. So this is how we're going to be able to control this movement along and kind of really get things happening and get things progressing so that there's lots of space for that new chyme that's coming in from the stomach. Great. All right, let's take a look at the chemical digestion in the large intestine then. Well, chemical digestion in the large intestine is going to be pretty minimal. Why is it going to be pretty minimal? Because we've already done it, right? We've done most of what needs to happen in terms of our chemical digestion in the small intestine way back when we had all of those enzymes, right? So we had the enzymes that were coming from the pancreas. We had the enzymes that were being secreted from the wall of the small intestine. So we've done that already. By the time we get here, we've done most of that chemical digestion, so there's nothing left to digest, and we don't really have any enzymes that are going to be secreted actively from the wall of the large intestine. We don't have another pancreas that's going to be releasing more digestive enzymes. We don't have another liver that's going to be releasing more bile for us. So we've kind of used up the amount of enzyme that we've got, and we've done most of the digestion anyway. So there's not going to be a whole lot that's going to be happening. So we're really only secreting mucus into that inner lining of the large intestine. But what we do have a lot of, as you know, are those bacteria that live in the large intestine, that live in the colon. And so a lot of times those bacteria are going to be maybe unpleasant for us, or give us some features that are a little bit unpleasant, but they do some really important jobs for us, as I'm sure you talked about with Dr. Stewart. They're going to be able to ferment some of our undigested carbohydrates. And when they ferment undigested carbohydrates, it's kind of like when you're making wine or beer or bread, you're going to get some gases that are released. And some of those gases that get released, right, that could be carbon dioxide a lot of times, it could be methane gas. Methane gas doesn't smell quite so nicely. Sometimes you'll have undigested proteins that are there, especially proteins that have sulfur in them, so things like cabbage and eggs that have some of that sulfurous compounds in it. You can make hydrogen sulfide gas. Smells a little bit like rotten eggs. When you get into the elevator with a person, and don't blame them, it's just their bacteria. They were not doing it on purpose. It's the bacteria that's living in their large intestine that is creating all of these gases and potentially these odors. So these are maybe not so helpful for us. Okay, if the bacteria are able to ferment some of those carbohydrates, break down some of those carbohydrates, if there's any that are free floating, the bacteria don't kind of immediately gobble up, well then you can absorb them as well. Realistically though, the bacteria are pretty greedy, they're going to eat whatever they need to survive. But what they do produce as waste products, a lot of times are some vitamins. So vitamin K, for example, is a waste product that some bacteria produce. Vitamin B12 is another vitamin as a waste product that they produce. And so we say, well, thanks very much, we'll let you live here if we can have those waste products. And so then we absorb vitamin K, we absorb vitamin B12. What do we need vitamin K for? Blood clotting, exactly, for creating some of our clotting factors. As we said, it itself is not a clotting factor, but it's helpful in the creation of clotting factors. B12, what do we use that for? Something with red blood cells. Yeah, so creating red blood cells in order to be able to generate the hemoglobin that we need vitamin B12. So again, these are pretty useful vitamins, and we rely on these bacteria to actually produce it for us. So we can get some of these vitamins in other places in our diet, but most of what we get is actually thanks to the bacteria that live in our large intestine. So yes, we put up with the gas and the odor to say thanks very much for those vitamins so that we have what we need. Right? In terms of the absorption, so we said there's not a whole lot of chemical digestion that's going on because there's no enzymes and there's nothing left to be digested that we can do on our own. We rely on the bacteria to do that for us. So this means that, again, there's not a lot of absorption that's going to be taking place because there's really nothing much left to absorb. There's a little bit of water, so that last kind of couple of 100 milliliters of water that we want to pull out. And most of it's absorbed in the small intestine, as we talked about a few minutes ago. Some of those last few electrolytes, so if there's any kind of last little bits of sodium and chloride that we want to be able to absorb, then we can do that. And then vitamins, obviously, that are produced by those bacteria that we just talked about. But other than that, there's not a whole lot. We're not talking about moving glucose. We're not talking about moving amino acids, any of those kind of nutrients. All of that has already been done. All right, so not much absorption is happening there. Let's talk about feces formation. This is what everyone wants to talk about at 10:30 in the morning on a Wednesday morning. Is let's talk about poop. Well, you're going to be becoming very familiar with poop in your profession. Poop is actually very, very important. Aaron talks all the time about poop at home, not ours, but other patients, because it's a very kind of key sign that they look for. What color is the poop? What shape is the poop? What's the consistency of the poop? How much are you pooping? Are you pooping enough? Or are you not pooping enough? So it's happening all the time. It's kind of like one of the key things that is limiting for getting patients out of the hospital. Have you pooped today? No. You can't go home until you poop. But doc, I just want to go home. Poop, and then you can go home. We got to make sure that your colon is working. Right? It's true, right? You have to be pooping. And so, what can help? Get up and walk around, right? The more you walk around, you get those muscles contracting, get the gravity working. Get that poop out, and then you can get home. So, you're going to be talking about a lot of poop. You're going to be seeing a lot of poop over the course of your practice, so you may as well just get it over with and talk about it now. The proper term for poop is feces. Really, and again, everybody's all grossed out, right? Feces, disgusting, is gross. Well, really, what is it? And people have the same reaction for some reason to urine. They're like, urine is gross. Okay, but when we talk about urine, all we're going to see is that it's mostly just the liquid portion of blood, right? It's just the liquid portion of blood with a couple of extra hydrogen ions in it and some urea. And then all of a sudden, urine's gross. Well, what is this? Like, 10 hours ago, it was your sandwich. Right? And you had no problem eating your sandwich. Right? And now you mix that together, you pull out the useful things, and you mix it with some bacteria, and that's essentially what feces is. So feces is just all of the undigestible components of the food that you're taking in, So things like cellulose, we're really not very good at being able to break down the cell walls of plants. It's very helpful in terms of fibrous material to be able to kind of scrape out the lining of your intestines and make sure that we're not having kind of collections of junk building up in there. So that's very helpful, but we can't actually do a very good job at digesting it. We're not cows. We don't have the proper enzyme to be able to chemically digest cellulose. So it just works its way through. It just stays there undigested. And so anything that you're not able to chemically digest is going to end up then just moving through that digestive tract and then is mixed together, of course, with any epithelial cells that are going to have sloughed off along the way. Of course, epithelial cells have a very high metabolic rate, so we slough them off the surface of our skin, we slough them off the lining of our gastrointestinal tract, and where does it go if it's in our GI tract? Well, it just ends up getting packaged together with all of the undigestible material of the food that we eat. And then, of course, there's going to be lots of bacteria there mixed as well, because you've got millions and millions of bacteria that are living there. Some of them are going to die, some of them are going to be alive, some of them will slough off along the way, and then all get packaged together into a nice little package that we call feces. So again, a lot of times people are like, it's so disgusting, but really it's just parts of your sandwich that you couldn't digest. It's bacteria, right? So how disgusting is that? All right, it's pretty disgusting. But let's talk about defecation. So defecation is the act of being able to remove the feces from your body. And it turns out that it's going to involve both a reflex as well as some voluntary action. So there's going to be a reflex that is involved that you have no voluntary control over. So we know that a reflex is always going to have two components. What are the two components to a reflex? Okay, back to last semester. What are the two parts of a reflex? Sensory and motor. So we need some kind of sensory information that's going back to the spinal cord, and then we're going to have some kind of motor activity in response to that sensory signal that's going to and then coming from the spinal cord itself. So that's going to be the reflex. There's going to be a sensory component and a motor component. And then there's going to be some somatic motor activity. That's your voluntary activity that you get to decide when some things contract or relax. So we'll start with the sensory part of our defecation reflex. So here we have our nice little distal end of the colon. So this is our rectum. And you can see that the first kind of trigger for this reflex is going to be the presence of feces in the rectum itself. So as part of that mass peristalsis that we were talking about a few minutes ago, and you're shoving kind of all of the contents of the colon or the distal ends of the colon towards that rectum, well, that's then going to force feces into the rectum itself. And the more you force feces into the rectum, well, the more the wall is going to stretch. And so you're going to have stretch receptors that are going to be found, just like in the stomach, you have stretch receptors in the wall of the stomach, you have stretch receptors in the wall of the small intestine to tell you, hey, there's some chyme in there. Well, now we have stretch receptors in the wall of the rectum to say, hey, there's some feces here in the rectum itself. And so those stretch receptors then are going to send that sensory impulse to the sacral area of the spinal cord. So there's our afferent nerve that we were talking about last semester, afferents are going towards the central nervous system to indicate that, hey, there's feces here, what do you want to do about it? Are you consciously aware of this at this point? No, you are not consciously aware. Nothing has gone up to the cortex of your brain to tell you, hey, something's going on down there. All we're saying is there's some sensory information from stretch receptors in the wall of the colon that are going to, or the rectum, that are going to end up in the sacral area of the spinal cord. All right, that's the sensory part of the reflex. Then we have the motor part of the reflex. The motor part of the reflex, again, does not involve the brain yet, so you do not have any voluntary control over this. We have that direct spinal cord reflex where we're going to have our parasympathetic nerve that is then going to signal for two things to happen. So pay attention to how this process works in terms of sensory part of the reflex, motor part of the reflex, and then a voluntary aspect that comes later. The same exact process is going to be taking place both for defecation that we're seeing here and for micturition, which is urination, that's going to happen as part of the urinary system. There's going to be a sensory part of the reflex, a motor part of the reflex, and then a voluntary component that is going to happen. So it's almost identical, just in two different structures, the rectum versus the urethra.
So in the sense
So the afferent nerves carry the sensory information about the stretch that's happening in the wall of the rectum to the sacral area of the spinal cord, yes. Great, that's where we're at, yeah. Okay, great. So then, once we have that sensory information that has arrived in the sacral area of the spinal cord, then we have the stimulation of the motor portion of that reflex, which is going to allow two things to happen. Again, not of your voluntary control. The first thing is that the internal anal sphincter is going to relax. What did we say that internal anal sphincter was made up of? That's our smooth muscle. So that's our smooth ring of smooth muscle, involuntarily controlled because it's controlled here by this reflex. And so it's going to cause that internal anal sphincter to relax, and that's what you can see right here. We're also going to see that the motor portion of this reflex causes the wall, the smooth muscle in the wall of the rectum, to start to contract. So now you've opened up a sphincter, and you've started to contract the wall of the rectum. So what that's going to do then, of course, is force some feces down into the proximal portion of the anal canal. So there's the feces that have been forced down now into this very, very distal end of the anal canal, and it's going to cause some stretching of the anal canal. Well, that now is going to be the trigger for you to say, oh, I gotta find a wash. I've got some feces going on, mass paracelsus just happened, I'm trying to make some space. So we're going to then send that information to the brain. So that sensory information of feces being in the anal canal, because we've relaxed the internal anal sphincter, we're contracting from the top, forcing some of that feces towards the outside, not outside yet, because what are you still controlling? The external anal sphincter, and you're hopefully keeping that nice and tightly closed, so there's no feces that's leaving yet, but you are at least consciously aware, because now that there's feces in the anal canal, there's going to be stretching of the anal canal. That stretch then signal is sent up to the brain, and then you say, oh, yes, I need to find a little pit stop, pull over to the side of the road, because it's time to avoid the colon. Now, does that happen immediately? As soon as the signal reaches your brain, do you have to relax that external anal sphincter? No. You can voluntarily keep it closed. In fact, probably one or two of you right now is saying we need to wrap this up and get to our break here, because I'm holding it closed for now, but who knows for how long? Because eventually, once you find an appropriate receptacle, well, now you can allow for that voluntary relaxation of the external anal sphincter, and now you've got an open pathway. So the internal anal sphincter is open, the external anal sphincter is open, the wall of the rectum is contracting, and so that's then going to force the feces to the outside of the body. You can help to increase the movement of this feces to the outside by contracting the abdominal muscles. That means you're just kind of putting extra pressure on the outside of the colon to help squish it to the outside, and then you're going to be able to essentially move the feces out of your body.
So that's like having a correlation maybe
Yeah, exactly. So when Aaron's at the hospital and the patient's saying, Doc, how do I go home? I just want to go home. Just get up and walk around. Because the more you walk around, the more you're contracting those abdominal muscles, you're squishing, essentially pushing on the outside of the colon, and that helps get things moving. And so again, if you're wondering what kind of is like the key for making sure that your colon continues to work properly, hydration, make sure that you're drinking enough water so that your feces don't get too dried out, and movement, get moving, and then that's going to help get that contraction happening and those feces to the outside. This is what feces looks like. You think, well, that's actually maybe not so scary. What is it? Well, it's all this undigested material, like all the plant, like the cellulose from all those vegetables that you're eating, all the different types of bacteria that are growing here. Obviously, this is a false color image, but all the different types of bacteria that are living inside your feces. And again, as we said, feces is made up of a mix of, yes, all the undigested stuff and some dead bacteria, but also live bacteria as well. It's estimated that half of the mass, if you want half of the weight of your feces, is actually contributed by bacteria. That's a lot of bacteria then that we find in these feces. So that's one stat. Half your mass of your feces is estimated to be bacteria. Another stat is that about a third of people don't wash their hands after they go to the washroom. A third of people. And you're like, oh, that's really expensive. Oh my gosh, that's not me. That's like, yeah, so it's like all of you, you don't, I don't wash my hands. Guilty as charged. Yeah, no, that's like all of you that aren't washing your hands after you go to the washroom. If you want to do a little creepy experiment, go stand in the washroom and just watch people come and go in the washroom. It's a little weird, but like watch and count how many people actually wash their hands. Like this morning, I went to the washroom, guy came in the washroom, walked right up, like, dude, what are you doing? Just wash your hands. Bacteria, even if it's like, you don't want to, it's gross, whatever, but it's just bacteria, just wash it off. There's so many bacteria, and from your microbiology class, like, wash your hands. What's the biggest thing that you can do? Wash your hands and don't touch your face, right? Just wash your hands, please. For the love of healthcare, just wash your hands after you go to the washroom. Okay?
I've seen
the
that
third of you that I know are out there that don't do it. Just do it. Wash your hands. Okay, we're going to take a 10-minute break. We come back.